April 27th, 2014, and my name is Darwin Bell, and in the Senior Physiology Lounge at the San Diego Convention Center. Today is my honor and pleasure to interview Dr. Gabriel Navar from Tulane Medical Center for the Society's Living History Project. Dr. Navar is a longstanding member and former president of the APS, who has worked in the areas of the intrarenal renin-angiotensin system, as well as the role of the kidney in hypertension. And as I said, an honor and pleasure to interview him today.
Thank you, Darwin.
Okay, Gabby, are you ready to answer a couple of questions?
Yes, Darwin. I'm very happy to do it. Good morning to all.
Can you tell me about your family upbringing and how you actually decided that you wanted to go into science?
Well, it was a very indirect way, and I think it reflects what happens when individuals that basically don't have any role models eventually go into science. I was raised on a farm, and we used to have a lot of activities going to ranches and riding horseback and herding cattle. And we had a dairy in El Paso, Texas, and I was raised in El Paso. And my ambitions were basically to take over my dad's job, and perhaps that I would be able to run the ranch or run the dairy. And I grew up raising cows and horses and chickens and having a lot of aspirations to go into agriculture. So when I told my dad I was going to go into agriculture and go to college, because my dad was encouraged, even though my dad didn't have hardly any education, I think he went to the 6th grade, he did want to encourage his children to go to college. So he really wanted the children to go to college, and I was the 4th in line, and I wanted to go and maybe perhaps major in animal husbandry or dairy husbandry. And my dad said, no, you need to do something like you need to go learn a profession when you go to college. So I said, well, maybe I'll go to veterinary school. And I ended up working for a veterinarian for some summers. And I enjoyed science in high school, so I thought, well, I'd enjoy science, but toward a veterinary profession. And I ended up going to Texas A&M after my high school years in El Paso. And I did that because that was the university at Texas that had the veterinary school. That was my ambition. And so as I finished up the pre-veterinary curriculum and went into veterinary school, I was really more and more drawn to the basic science courses. And while all the other members of the class were just eager to go into the clinical years, I really enjoyed the basic science courses. And I found out I really liked physiology. So I decided to maybe instead of going out and be a practicing veterinarian, I might go into veterinary education, and so I thought I needed a PhD. And at that time, Dr. Arthur Guyton had a program for graduate students in the University of Mississippi, which seemed like a far, far away place for me. And so I ended up going to the University of Mississippi to get a doctoral degree with Dr. Guyton. And I took a leave of absence from veterinary school. I never went back, but I had my bachelor's degree. And so then I went on and got my PhD with the very, very highly recognized and well-known scientist Arthur Guyton, who was professor and chairman of the Department of Physiology there in Jackson, Mississippi at the University of Mississippi Medical School.
You know, I started working with you in Jackson, and it was quite an honor to be in Dr. Guyton's department. What influence did he have on your career and on your training?
Well, he was a very, very interesting individual, and of course he was very well respected and an international expert in physiology. His textbook has been translated into over 20 different languages, and he was well known and highly respected throughout the world. But he really spent a lot of time with his students, with graduate students and his postdoctoral fellows. I'm actually one of the few individuals that did both, got both the graduate degree, my PhD from Arthur Guyton, and I did my postdoctoral work with Dr. Gaiden. He was very challenging. He would always challenge you to explain the mechanisms by which things, your physiological processes occurred. And so he really always encouraged us to go for the unknown, try to explain the mechanisms, and he spent a lot of time with his fellows. He personally taught some of the graduate student courses, and it was really a lot of fun to work there. And it was a challenging environment, and it was quite interesting. And it was those were interesting times too in Jackson, Mississippi. Those were quite turbulent years for a number of reasons, but most of us that were there in graduate school, we just kept churning away in graduate school and didn't pay attention to many of the political upheavals that were going on at the time. But it was an exciting period, and I learned a lot from Arthur Guyton, and I often will quote him, like, explain the mechanism, show me the data, and things like that. It was always very exciting to work with him. And it was a lot of fun to work with him. After I finished my graduate degree, I stayed on as a faculty member for a few more years, but then I realized I did need to have some exposure to other laboratories and other techniques. And so I spent a year as a special postdoctoral fellow at Duke University. And at Duke University, I had the opportunity to interact with Ike Robinson, Jim Clapp, and several other members that were there in the Raleigh-Durham area. And I also got the chance to know Dr. Carl Gottschalk very well at the University of North Carolina. And that was a very, very productive year for me. That's where I learned to do micropuncture. And when I went back to Jackson, Mississippi, I set up the micropuncture lab. And that was about the time that you came and joined us and started working in our laboratory. So that was also a very exciting time, because we were able to use our new micropuncture techniques to establish some important mechanisms related to autoregulation of glomerular filtration rate and glomerular pressure.
So how did you develop an interest in the kidney? Because Guyton was a kind of a whole body physiologist, and how did you develop that?
That's right.
Interest?
That's right. It's interesting. Another area that's very indirect, and that is, I don't think hardly anybody starts out deciding they want to be a kidney physiologist, because it's an area that sometimes is not quite that attractive. And I actually thought that I was going to be in neurophysiology. I told Dr. Guyton when I went there that I really was interested in neurophysiology. And he said, okay, that's all right. We have a good neurophysiology program. I've always enjoyed neurophysiology. And he gave me a book, and he said, hey, here's a textbook. In there, it tells you how to develop a diabetes insipidus model. And I need a diabetes insipidus model that doesn't have any antidiuretic hormone, so that I can study what's happening in the kidney. Because he was very interested in learning more about the role of the kidney in the control of blood pressure. So I spent a lot of time doing surgical experiments, learning to do diabetes insipidus models in dogs. And at that time, we had access to mostly large animal experiments. And we prepared the diabetes insipidus animals. And then when he said, well, I'll have to get somebody else to do those experiments, because you want to, because they're kidney physiology experiments. He said, no, those are my studies. I'm going to do those experiments. So I started studying the changes in autoregulation and the changes in sodium excretion and water flow and urine volume that resulted when the animals didn't have any antidiuretic hormone, because I had personally done the surgery that had transected the pituitary stalk, and so I was able to get diabetes insipidus preparations. And then I started studying kidney. So then I started studying autoregulation of blood flow. I started studying pressure natriuresis and changes that occurred. And little by little, I got more and more interested in the studies of the kidney and the mechanisms of blood flow autoregulation and the changes in sodium excretion that occurred with changes in blood pressure. And so I became very interested and fascinated by not only the control of renal blood flow and glomerular filtration rate and sodium excretion, but also how the changes in kidney function would actually influence the development of hypertension in this model. So little by little, we went and became more and more involved in the role of the kidney in the control of blood pressure, the role of the kidney in either acute or progressive hypertension. And of course, that brought in a lot of interest in the renin-angiotensin system. And I was influenced a lot by a very prominent physiologist, endocrinologist, that was also there in Jackson, Mississippi, by the name of Herbert Langford. And he really encouraged me to learn more and more about the renin-angiotensin system. And way back there, we ended up writing a chapter together on the effects of angiotensin II on the kidney. So all that together ended up being an area of science that became my turf, and it's one of the areas that we've continued to work in and to learn more and more about these mechanisms by which the kidney can influence the control of blood pressure.
So I know you've worked in a number of areas and that you've had and published a number of very, very important studies, and this may be a little unfair, but what do you think your most important contribution to science has been?
Well, yeah, you're right. That's a little hard to say, but clearly one of the most important studies that we ever did was a study that we published in Science together. You were the first author, and we published in Science that there was a potential role for the intracellular calcium in the macula densa cells to help to regulate the tubular feedback mechanism. I personally think that that's still a very valid area of research, and I think that the publication in Science was a really highlight for our work, and we really enjoyed that study. So I think that's a hallmark study in relationship to the tubular feedback mechanism. I think the study that we did and published in Circulation Research about the effects of blood pressure on sodium excretion was very important. And more recently, I think the work that we've done with the role of the intratubular renin-angiotensin system has become very, very prominent area of research for us. And we think at this point that clinically, from a standpoint of clinical implications, that's probably the most important work, where we have shown that in angiotensin II dependent hypertension, in other aspects of hypertension, there is augmentation of the intratubular renin-angiotensin system, where the proximal tubule cells are stimulated to produce more angiotensinogen, and the angiotensinogen is secreted into the tubular fluid and forms more angiotensin II, not only in the proximal tubule, but also in the distal nephron segments, and that stimulates angiotensin II production, which augments sodium transport and contributes to the long-term effect of angiotensin on blood pressure. So I think those are very, very important studies, and I believe these probably have the most clinical translational implications.
So I know that mentoring has been a big part of your career. Would you like to make a couple of comments about your role as a mentor?
Oh yes, that's been probably the area that has been the most rewarding for me. Being able to start with individuals who just either got out of college or got out of medical school or entered graduate school, and being able to help them develop into scientists, that's been an extremely rewarding area. I've had the opportunity to mentor many, many different PhD students, postdoctoral fellows, both PhDs as well as MDs, and this allowed me to have interactions with people from all over the world. I have former students and postdocs that are in China, in Taiwan, in France, in Poland, in Hungary, and many other areas, in the Netherlands, for example. So it's really been one of those extremely rewarding areas, and I think it's the most fulfilling of all, the opportunity to help young people develop. Just as Dr. Gaiden helped me develop, and Jim Clapp and Carl Gottschalk helped me develop into a better scientist, I've been able to help other young people. And so I feel very good about that, because almost all the individuals that have trained with me have had very good experiences with us, and we've maintained very close friendships. And everybody that's ever worked with us, alumni, I call my physiology alumni, and we've had maintained good relationships and good interactions throughout the years. And that isn't always true. There's a lot of individuals that do not have fond memories of their postdoctoral years or their graduate student years, but I think most of our students. And of course, you were probably my first real graduate student, and I was really, really proud of the way that you developed from a research assistant into a graduate student, postdoctoral fellow, assistant professor, always looking at greater and greater opportunities and greater challenges. And now you're an endowed professor at University of South Carolina Medical School, and it's very, very rewarding to see people that have been associated with me grow. And I think I've benefited more from my interactions with all these young minds and students and postdocs than they have. It is I who have really benefited the most from the many interactions we've had with the students. So mentoring has been a very exciting time, a very exciting aspect of my career.
Yeah, so you can certainly see the number of people that you trained around the world, really, and how well they've done, and
Yeah,
it's just amazing.
That is important. It's important to see some of the people that you train actually go on and become prominent individuals. A lot of folks will train postdocs and then find out that they kind of disappear from the scene. And while I've had that happen as well, I'm very proud of the fact that many of the people that have trained with me have gone on to very prominent positions, not only in the United States but throughout the world.
So tell us about your first experimental biology meetings, and how it differs between now.
Yeah, that was a long time ago. I remember most fondly the fact that we used to have fall meetings. The American Physiological Society used to have 2 meetings a year. One was the big meeting, which almost always occurred in Atlantic City. It was just like this experimental biology meeting we're at here. And these were experimental biology meetings with all the various societies. But they almost always had them in Atlantic City. That was before Atlantic City became a gambling mecca. And those were very good meetings, and they're pretty much the way they are now. But the fall meetings were really very, very pleasant because they usually had less than 1,000 people that participated in the fall meetings. It was all Physiology Society, and we had an opportunity to meet and talk with many of the prominent physiologists that were working in the area. And I can remember going to these sessions where there may be only 20 or 30 renal physiologists, and many of them were very, very prominent physiologists like Dr. Pitts and Dr. Windhager and Dr. Gottschalk and many others. And at those small meetings, we had the ability and the opportunity to interact in a way that was very, very rewarding. So those were good. I can remember, perhaps the one I remember the most is a little meeting we had in Bloomington, Indiana. I'm not sure it was the very first APS meeting I went to, but it was certainly one of the first. But then later on, the American Physiological Society stopped having the fall meetings, and we just have the one big meeting every year. But we've also had the FASEP summer conferences where we bring in maybe only 100, 120 people that are interested in the control of the kidney, and those are very rewarding as well. They have been a lot of fun because we have a chance to interact with a small group of people.
So I know your family has been important in your career, in Randa and your children.
Yeah, sometimes I feel guilty about that because I think scientists sometimes are married to their science more than their family. And so it's been important to balance it. I have 4 children, and my wife Randa and I have been married almost 50 years. I think next year it'll be our 50th anniversary. And so we've hung together, and she's been very supportive. But sometimes I feel that I call them science widows and science orphans because you sometimes have not had as much opportunity to spend time with the family as you would like to. And so it's been a little bit of a challenge, but I think that we've been able to maintain a very, very good interaction with all the family, and now all the kids are grown and living, but we keep very good interactions. I have 2 boys and 2 daughters and 3 grandchildren, and my wife has remained very supportive of all the work that we do, and that's been very rewarding. But sometimes in science, you just don't have as much time to spend with the family as you'd like to.
Well, I want to thank you, Gabby, as my mentor and colleague and my friend for participating in this living history program for APS, and I wish you many more years. And
you know, I think we probably should mention a little bit about how we got involved in the leadership of the American Physiological Society, because that was an area that developed also kind of slowly. It was clearly, I was very interested in the American Physiological Society, and I was really eager to participate as much as possible. And I got a chance to know the members of the Physiology Society, and we formed the renal section. When we were forming the various renal sections, we formed the renal section. I was involved in starting the renal section of the Physiology Society. And so so through the years, I developed more and more interest in leadership opportunities with the American Physiological Society, and I was able to get on the council, get elected council. I served as associate editor for American Journal of Physiology for many years, because I did it both with when Tom Andreoli was the editor, as well as when Jim Schaefer was the editor. And then I became involved both in the chairman's group after I became chairman at Tulane. I became involved both the chairman's group, and so it's been very rewarding to be able to grow in leadership opportunities. I served on council, and then I became president of the chairman's group, and I was also president of the American Physiological Society. So we've had a lot of fun during those years trying to serve in leadership roles in both the American Physiological Society as well as the chairs of the departments of physiology, where continues to be a very important area for us.
I know it's played a very important role in your career, and you certainly have made major contributions to the American Physiological Society.
Yeah. Okay, well, thank you very much, Darwin. It's been a real privilege and an opportunity to be involved in this interview, and I look forward to further discussions.
Thanks. Thank you.